Hi student, today we are going to see about bacterial motility. Definition of the motility. Motility is the ability of a cell or organisms to move of its own accord by expending energy. Motility is closely linked with hemotaxics. Hemotaxic means movement of organisms. The ability to orient along certain chemical gradients. A large number of bacteria are motile in nature. The most possess one or more flagella on the surface that allow them to swim. Importance of bacterial motility, hemotactic behavior and survival. Ability to change the direction, that means moving away or toward repellents or attractants, and avoid unfavorable condition of habitat and choose favorable environment. Next, pathogenesis for attachment and colonization of cell wall of the host cell. Types of movement. There are three types of movement: flagellar movement and gliding movement and Brownian movement. Flagellar movement. Most motile bacteria move by use of flagella, thread-like locomotor appendages extending from the plasma membrane and cell wall. Next, gliding movement. It is a sliding movement without any appendages. That means without using flagella. Movement without any appendages. Next, Brownian movement. It is a random or zigzag in direction. Movement exhibited by particles suspended in liquids due to the bombardment of water molecules. For example, Saccharomyces cerevisiae and Staphylococcus aureus. Structure of flagella. They are slender thread-like appendages protrude from the cell wall. It measures up to 15 or 20 micrometer in length and 0.01 to 0.02 micrometer in thickness. It is made up of one special protein called flagellin. The fine protein structure requires special staining technique for demonstrating them with the light microscope. That special staining is tannic acid staining, Leifson method and Roos method. Arrangement of flagella. Flagella vary in number and arrangement. They may be polar at one or both end of the bacterium or lateral along the sides of bacterium. Monotrichous, lopotrichous and peritrichous and amphitrichous. First one, monotrichous. Monotrichous means some organisms have a single polar flagellum. This condition is known as monotrichous. For example, Vibrio cholerae and Pseudomonas species. Next, lopotrichous. Some organisms possess cluster of flagella at one end. This condition is known as lopotrichous. For example, Pseudomonas fluorescens and spirillum. Peritrichous. Some organisms possess flagella all around the body. This condition is known as peritrichous. For example, Salmonella typhi and E. coli. Next, amphitrichous. Some organisms possess single or cluster of flagella at both ends. This condition is known as amphitrichous. For example, Aquasperillum serpens and Alganensis faecalis. Next, the ultra structure of flagella, filament, basal body and hook is the important structure of flagella. First one, filament. The longest and most obvious portion is the filament, which extends from the cell surface to the tip. It is made up of a special protein called flagellin. Next, basal body. A basal body is embedded in the cell. The basal body attaches the flagellum to the cell wall and plasma membrane. It is composed of a series of rings connected to the central rod. It working like a motor. Next, hook. A short curved segmented. The hook is present outside the cell wall and connect filament to the basal body. The shown ultrastructure of flagella for gram-negative and gram-positive bacteria. Next, mechanism of flagellar movement. The filament is the shape of the ring helix and the bacteria move with the helix rotates. The basal body acts as a motor and cause rotation. Flagellar rotation determines the nature of bacterial movement. Next, detection of bacterial motility using various methods. First, directed demonstrating of flagella and indirect demonstration of motility. First, direction demonstration of flagella, tannic acid staining, Leibson method and Roos method, and electron microscopic method. Indirect demonstration of motility, Griggs tube method and hanging drop method, and semi-solid medium, dark ground or phase construct microscope. First, direct demonstration of flagella using wet mount staining method. Stain, when we are applying stain, it stain the flagella like a purple in color. Next, Leibson staining method. Adding modern tannic acid to increase the thickness. That means protein colloidal precipitate with the dye that absorb into the flagella. Flagella look like a thick structure to will appear in light microscope. Next, Griggs tube method. It is a most common motility test. The inoculation made inside the inner tube and subculture is taken from the surface of the medium in the outer tube, whether it is motile or non-motile. Hanging drop technique. Preparation of the cover slip by dapping petroleum jelly on the corner and place loop of microbial suspension, that means culture, into the center of the cover slip. Turn the concave depression glass slide upside down over the cover slip. Invert the concave depression glass slide to produce hanging drop the specimen and to observe under microscope. Thank you.